Good afternoon, everyone. Welcome. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present and emerging. Welcome to the September FightMND Scientific Seminar. My name is Dr. Sarah Bennett and I'm the Research Coordinator at FightMND. Last month, we heard from Dr. Sam Barton from the Florey, who took us through her research into induced pluripotent stem cell models, oligodendrocytes, and how she uses these to understand myelin dysfunction in MND. If you missed out on this talk, it's now available on our website, so you can catch up at a convenient time. Following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you are using the app-based version of Teams and unfortunately doesn't work if you're watching on the web-based version. Please make sure you do introduce yourself and where you're from before asking your question. So now to our speaker from today. Dr. Rebecca Sangil is a postdoctoral fellow in the Neurodegeneration Pathobiology Lab, situated at the Queensland Brain Institute within the University of Queensland. She completed her PhD at the Illawarra Health and Medical Research Institute at the University of Wollongong in 2018. As part of her doctoral studies, Dr. Sangil was awarded the Endeavour Research Fellowship to conduct some research at the University College of London. Upon completing her PhD, Dr. Sangil was awarded the inaugural FightMND Early Career Research Fellowship to continue her MND research journey under the mentorship of Dr. Adam Walker at the University of Queensland. And she joins us today to present some of the results from that fellowship, creating a molecular map of proteinopathies in the MND brain. So thank you, Rebecca, and over to you.
Thank you, Sarah. Okay, how does that look, Sarah? Looks good?
Yep, all good.
Great. So thank you very much, Beck, Sarah and FightMND for this opportunity to present my research. I'll be sharing with you our latest results that we recently posted to bioRxiv, where we looked at the molecular signatures of motor neuron disease. And specifically, we were interested in trying to better understand this progressive loss of upper motor neurons in the cortex and interested in the effects of protein misfolding in the disease. So in our lab, we focused on a protein called TDP43, and this is because they're found, or inclusions of this protein are found in 97% of MND cases, illustrated here in Rachel Tan's review back in 2017. This is what TDP43 inclusion looks like in a motor neuron in the motor cortex of a stage 4 MND patient. So it sits in the cytoplasm of a neuron, and it's labeled here with phospho-TDP43, which is the marker of a mature inclusion. So the protein itself has many different functional domains, and illustrated here, we have a nuclear localization sequence, RNA recognition motif 1 and 2, and a glycine-rich domain. And these different functional domains enable TDP43 to affect its RNA and DNA binding activities in the nucleus of cells. And this is an essential protein, so we require this protein to be functional in the nucleus of all cells at all times. However, in motor neuron disease, it slowly becomes dysfunctional over time. So here I have a schematic of a motor neuron, and this is the nucleus here, and this is the cell body. And TDP43 progressively is lost from the nucleus. So we have this functional loss of TDP43. So it's not in the right place in the right time anymore, and it starts to accumulate in the cytoplasm. And here in the cytoplasm, this is where TDP43 becomes extensively post-translationally modified. So it will be modified with phosphorylation or acetylation. It becomes fragmented and truncated. And these different modifications lead it to become progressively misfolded. And misfolded TDP43 forms inclusions. These inclusions are very strongly correlated with the degeneration of motor neurons as well. So we believe by investigating this process of TDP43 dysfunction, we will take a step closer to understanding how the disease is initiated and maybe how we can stop disease. So what are the consequences of cytoplasmic TDP43 in the cortex? So here I have illustrated the time course of disease with this arrow. Here's the pre-onset phase of disease, where I've got a question mark here because the pre-symptomatic prodromal period could be years or it could be decades, and it can vary from person to person. Onset represents the onset of muscle weakness and wastage visible at the doctor's surgery. And early disease is where symptoms are already in play and progressing, and late disease and end stage are, I guess, the life-limiting end of the disease. What I thought I would do was to place what we know about the molecular understanding of the disease at each of these different time points. So at onset, we know that there's an increase in the mediators of the integrated stress response and a decrease of mitochondrial respiratory chain complex proteins. In early disease, we see a decrease in the number of synapses and a decrease in synaptic proteins, the beginnings of inflammation and microglial activation and dysfunction and astrogliosis, and neuronal hyperexcitability. And by the late stage of disease or end stage, we have worsened inflammation and microglial activation and astrogliosis. We have fewer synapses, fewer mitochondrial proteins, and fewer microtubule proteins. So those are the proteins involved in maintaining axonal integrity. And then the last one I want to mention is this decrease in proteostasis components. And these proteins are also sequestered to TDP43 inclusions. So combined, there's generally a decrease in the functional pool of proteostasis components. And we reviewed this recently in Progress in Neurobiology. So we really know quite a lot about the disease at the end stage because we have access to postmortem human tissues, but really we have an emerging picture of what's happening at the front end of the disease. And I believe that the snapshot of the molecular signature of end-stage tissue might not necessarily reflect what biological processes are underway at the early stages of disease. This leads us to, I guess, a problem in the field is how do we study the early stages of disease if we don't have access to tissue? And the way in which we went about that is to employ a mouse model of TDP43 disease. So this model was developed by the boss, Adam Walker, during his postdoctoral years. And what he did was genetically modified these mice so that they have a doxycycline regulatable human TDP43 delta NLS transgene. And what that means is that the mice after birth are maintained on a feed with doxycycline, and that suppresses the transgene from being expressed. So the mice are normal. But as soon as the doxycycline is removed from the diet, it results in the expression of this transgene, which is a cytoplasmic localized TDP43. And what happens is basically progressive motor deficits and behavioral dysfunctions that look like ALS or model ALS and MND in the mouse. And at the molecular level, what we see at onset, which is a 2-week time point, here we have a control mouse showing nuclear TDP43 in the neurons. Neurons are labeled with neurone in green. And here in the RNLS8 mouse, that we've got cytoplasmic TDP43 in the neurons. So I guess the model looks as it should. So when the mice are tested by a rotarod, it's basically a rapidly spinning bar, which assesses the mouse's ability to, I guess, have motor deficits. Over time, as the doxycycline is removed, you see a very dramatic decrease in its ability to undertake this rotarod test. And at 6 weeks, it can no longer run on the bar. And these mice also show a sharp decrease in survival as well. A really useful feature of this model is that the doxycycline can be reintroduced, so you can functionally recover these mice. So here's an example of where if you reintroduce doxycycline into the diet, you stop the transgene from being expressed, the mice functionally recovered and they're able to perform really well on the rotarod again. And they actually survive similar to controls. So this model represents a really useful tool to start to study this question. So what are the consequences of cytoplasmic TDP43 in the cortex? And also, it allows us to investigate this aspect of recovery. So we can have a look at the proteomic signatures or the molecular signatures of all of these different disease stages, but also recovery, which is really exciting. So back when Adam was in Macquarie, he initiated this collaboration with Medi to conduct a longitudinal proteomic study of the cortex of the RNLS8 mouse. And what they did was they collected a cortex, or partially collected the cortex at pre-onset, onset, early disease, late disease, and in recovery. So we have this really detailed timeline of, I guess, the proteomic signatures of each of these time points. So the individual samples were labeled with a TMT label, and each of these is a unique label, and then they're pulled together and then they're analysed by mass spectrometry. And then the data set, the resulting data set, was then analysed by Dana and Gemma, who were at Macquarie at the time. So this data set is really rich and very detailed. So there's a lot of results that came out of this one experiment. And here's just an overview of those results. And just going to use this slide as a landmark so we can progressively work through the findings. But firstly, I want to share with you the first longitudinal map of the cortex proteome throughout a TDP43-driven disease. And it looks like this. So here we have, I guess, a very high-level overview of all of the proteomic changes that occurred in the cortex of these RNLS mice through time. So this is the protein abundance ratio, and proteins that are increased are in red and proteins that are decreased are in blue. And here on the left we have control, and on the right we have the RNLS, the disease mice. And then each column represents a different time point, each row a different protein. So taking a look at this heat map, I think you can gain an appreciation that there's a very clear subsets of proteins that are increased in disease, so just here, and then a very clear subset of proteins that are decreased in disease, and then a variation of many different combinations in between. So we're going to go into more detail into each of these changes over the next few slides. So we had a look at the principal component analysis of these samples and found that PC1 clustered the control samples over here to the right and the disease samples here on the left. So PC1 distinguishes genotype. PC3 distinguished each time point. So here we have the disease mice at pre-onset, onset, early disease, late disease, and recovery. And there's this really beautiful stepwise migration of each of these samples through the PCA plot, showing that they are all distinct samples. And here what I think is interesting is that this subset of samples, the recovery samples, start to migrate back towards control, but after 2 weeks back on docs, they're not exactly like controls. They have distinct proteomic signatures. They're also distinct from the sample at onset as well. This plot here on the right shows the number of significantly increased and decreased proteins at each time point. And what you can appreciate is that there's an accelerating increase of the number of proteins that are significantly altered at every single time point. And another interesting finding here is that in recovery, the number of proteins that are altered partially are restored back to control levels. But there still are unique alterations in recovery as well. So one of the different ways that we analysed this was using a pairwise comparison. So we took the cortex proteomics of each time point and compared it to control. And this volcano plot in the centre shows us each individual protein and its log fold change and its significance. So here on the left are all the proteins that were significantly decreased, and then here on the right are all the proteins that were significantly increased in this box here. And we applied gene ontology analysis to these groups of proteins to see, what are the proteins that are enriched and what biological processes are they enriched for in each of these different groups. And at this onset stage, 2 weeks off docs, we found that there was an enrichment for membrane depolarization and neuron projection morphogenesis. And those proteins involved in neuron projections are highlighted here in blue. And regarding those proteins that were significantly increased, we found that they were enriched for lysosome, protein processing in the ear, and also one of interest to me was protein folding. And those associated with protein folding are highlighted here in red. The other thing that I highlighted here on this volcano plot are proteins that have been previously associated with ALS or MND. And I guess the obvious one is TDP43. So this shows the increase of the transgene at this time point. And then the other ones we looked at were TTBK1, which is a kinase that phosphorylates TDP43, ataxin-2, Stathmin-2, and UNC13A. And I picked these because they've been highlighted recently in some high-impact articles. And one of the reasons I highlighted these two in particular was that we found them to be altered throughout the time course of disease in the mouse model. So this sort of view of what are the proteins that are changed in this time point is kind of a very static snapshot of the processes that are altered in disease. And we have this information for each stage of disease that we collected, but it doesn't really give us an idea of the factor of time. So we analysed the data in a second way. So we used a different approach to be able to really visualize the dynamic changes to the cortex proteome that we could see was happening. And to do this, Dana applied weighted correlation network analysis of this longitudinal quantitative proteomics data set and showed that there were different unique subsets of proteins that correlate with different disease stages. So here is a network heat map of all of the proteins that were assigned a module. And this basically is a heat map showing the similarity score and the relationship of one protein to another, and that fell into this module here. So all the modules are named at different colours. So this one, for example, is the turquoise module, and Dana identified 8 distinct modules, and 5 of these modules showed disease-related changes in protein abundance over time. So here are those 5 disease-relevant modules. So firstly, I just wanted you to sort of zoom out and look at that whole slide as an overview. What I wanted to point out was that each of these protein abundance curves show different dynamics, different waves over time of disease. So for example, here we have one module that's going up and stays up in recovery, and here we have a module that goes down and then is partially reversed in recovery. So this type of analysis was able to sort of integrate time and show us that there are dynamic waves of both biological process activation and depletion throughout the time course of disease. So we'll take a look at this first module, the black module. Here, I'll just sort of go into more detail into this plot. We have the control samples that show pretty much a steady state over the time, so over week 1, 4, and 6, and in recovery. And then here in the disease half of the plot, you can see there's a stepwise increase in the abundance of these proteins. So this protein set had 38 different proteins in it. And what we did was subjected each of these protein sets to gene ontology enrichment analysis to understand what are the biological processes that are enriched in these modules. For the black module, we found that it was enriched for proteins associated with ensheathment of neurons, otherwise known as the process of myelination. For the blue module, this was associated with TDP43, and the proteins in this module were also associated with transport. That was one of the top significant processes. The brown module was catabolism. The turquoise module was enriched for neuronal systems, that's chemical neurotransmission. Here you can see that it's steady in control, and then it decreases progressively throughout disease. And what's interesting here is there's a partial normalization back to control levels in that recovery time point here. And lastly, this magenta module was enriched for protein folding factors. It had 23 proteins, and to me, when I first saw the data set, it was one of the most interesting modules because it shows no change in controls, but then a really dramatic increase at pre-onset, onset, and then a very dramatic normalization back to control levels in early disease, late disease, and in recovery. So I thought this was a really interesting module that might be representative of protective or factors that are increased in response to cytoplasmic TDP43, but cannot be sustained, so they go back to control levels throughout disease. So these modules also presented a very nice opportunity to investigate the cell autonomous and the cell non-autonomous contributions to disease. So we subjected those modules also to a cell type enrichment analysis, where I basically took protein sets that were in the literature already known to be associated with astrocytes, oligodendrocytes, microglia, and neurons, and compared those with the protein sets of each of the modules. And what I found was that the black module showed one of the strongest enrichments for oligodendrocytes, and the brown module was associated with microglia, and the turquoise module associated with neurons. So the remaining two disease-specific modules, the blue module and the magenta module, showed no cell type specificity. So I'll just go back to this previous slide and then annotate with the cell type that is responsible for these patterns of protein expression of these groups of proteins. The black is representative of oligodendrocyte proteins, the brown of microglia, and the turquoise of neurons, and magenta and blue did not have any specific cell type specificity in their protein lists. So like I said, this magenta module was a very interesting module for me to further explore. So we'll have a look at the different proteins that were involved in this magenta module in more detail. So here we have the list of all the 23 proteins that were, that displayed this protein abundance profile through time in disease in the magenta module. And you can see there's this really strong increase at pre-onset and onset of these proteins. And looking at the gene ontology analysis, you can see it's enriched for HSP90 chaperone cycle, protein folding, and regulation of protein binding. And there was an existing or a protein-protein interaction network that was based off previously published work connecting 7 of these proteins to one another. So it made me think, these proteins are here at the same time. Are they there in the same place? And could they be working together with one another to execute their activities in protein folding? So we took a look. We did some immunofluorescence of the primary motor cortex to take a closer look to see where are these chaperones? So I'll just go back one slide. We focused in on these four: calreticulin, HSP90, AB1, CDC37, and DNAJB5. So the way that this slide is laid out, we have control on the top and RNLS on the bottom, and then control on the top and the RNLS disease mouse on the bottom again. And I think the most striking result was this result with DNAJB5, where you can see that there's a really strong increase in the cytoplasm of neurons, of TDP43 positive neurons specifically, of DNAJB5 in the cytoplasm. So it's at the right time at the right place. Here with HSP90AB1, you can see a more modest increase. CDC37 shows an increase in the cytoplasm of neurons, but also more generally in the background as well. Calreticulin also shows an increase in the cytoplasm of neurons as well. So all four of these are increased in the cell bodies of neurons. So even though there was no specific cell type enrichment for the magenta module, the protein folding factors of interest in that module were increased in neurons with TDP43 in the cytoplasm, which is interesting. So the next thing that we did was to sort of drill down a bit more and focus in on the mechanism. So if these proteins are increased, why are they there? What are they doing basically? So we focused in on one of these protein folding factors. So we focused on DNAJB5, and we asked, I guess, the very broad question, is TDP43 a client of DNAJB5? And the reason that we focused on this protein is that it showed the highest fold increase at pre-onset and onset out of all the chaperones. And it showed a disease-related increase in the cytoplasm of the TDP43 positive neurons, as I showed in the previous slide. It also showed a co-localization with TDP43 puncta in the motor cortex of the RNLS mice, so not shown here. And also the relationship between DNAJB5 and cytoplasmic TDP43 has not been previously studied. So it felt like a good opportunity to investigate what is this protein doing. So the first experiment that we ran was to just very simply co-express TDP43 mutant, that's a cytoplasmic mutant, with DNAJB5 or a negative control. And what we saw in the insoluble protein content was significantly decreased. So this is TDP43, it's significantly decreased compared to controls. And then we looked at the phospho TDP43 at serine 403 and 404. It's a marker of more mature inclusions, and we found a significant decrease in the DNAJB5 samples as well. And then likewise, a decrease in the phospho TDP43 marker at the serine 409 and 410 sites. So this is the quantification here, and the most dramatic decrease was an 80% decrease in the insoluble phospho TDP43 at the 409, 410 serine sites. So we have here, I guess, our first indications that DNAJB5 significantly decreases TDP43 aggregation. But we wanted to take a closer look in neurons. So we conducted experiments with primary cortical neurons and co-transduced the neurons with the same cytoplasmic mutant and the same control. So the control versus DNAJB5. And here in this image, the TDP is in green, and each of these yellow arrows points to a TDP puncta in the cell body. And you can see there are many more puncta in this cell body compared to these cells here, which have the DNAJB5 co-expression. So there's 3 neurons here. And this is that quantified, so there was a significant decrease in the number of puncta per neuron, and there was a decrease in the percentage of neurons with puncta as well when we had DNAJB5 overexpressed with cytoplasmic TDP43. So the last thing we wanted to look at was if TDP43 is truly a client of DNAJB5, it will form a stable interaction. So The way in which the HSP40 family of proteins work is that they connect with the misfolded protein and then they deliver that misfolded protein to HSP70 for either refolding or degradation at the proteasome. So DNAGB5 was co-expressed with cytoplasmic TDP43, and then we conducted, or Shilpa, a master's student at the time, conducted these immunoprecipitation experiments where she pulled down GFP, and along came DNAGB5 flag and also HSP70. To be very thorough, she also conducted the inverse immunoprecipitation using the flag. So she pulled down DNAGB5, here it is here, and sure enough, TDP43 and HSP70 came down as well in those experiments. So this gives us evidence that cytoplasmic TDP43 and DNAGB5 are forming stable interactions, and they're also interacting with HSP70 as well as part of the refolding or degradation machinery. So we have a lot of data here on DNAGB5 as a protein, as an anti-aggregation protein. And what we wanted to explore further were, I guess, some experiments in the inverse setup. So here we've overexpressed DNAGB5. We now wanted to explore what happens if you knock DNAGB5 out. So is there any redundancy in the system? So what we did was we brought in from Jackson Labs the CRISPR DNAGB5 knockout mouse line, and all of the CRISPR DNAGB5 knockout mouse work was led by this team here. So Heleth, Lydia, Kit, and Juliana. And I guess we first proposed that if you knock out DNAGB5, there was probably going to be changes in the levels of TDP43. So the first experiments that we did were to take the cortex of these DNAGB5 knockout animals and to run proteomics on those and see whether or not TDP43 was altered. And if not TDP43, what other proteins were altered? So here's the summary of the proteomics from the DNAGB5 knockout cortex. We have log-fold change and p-value, and here's DNAGB5 significantly decreased, and here's TDP43, so there's no change. So that was, I guess, not what we expected, but that was the result. But we also found that there was a decrease in 82 proteins and an increase in 119 proteins. And out of those that were decreased and increased, we found quite a few of those were actually related or associated with motor neuron disease. So here's one, FA4, that was decreased. And here's HNRNP-U, HNRNP-A1, FUS, and MAPT or tau that were increased. So the CRISPR knockout of DNAGB5 in the cortex regulates levels of other MND associated proteins in the cortex. So it seems to be quite a crucial protein for the regulation of these proteins. We next wanted to evaluate whether in the presence of cytoplasmic TDP43, whether DNAGB5 knockout had any effect. So the team set about breeding wild type, heterozygous and homozygous DNAGB5 knockout animals, and then injected those animals with TDP43 delta NLS, so with the defective nuclear localization signal, or with a GFP control AAV. And they did this by ICV injections at P0. And then they conducted routine weighing and neuroscoring, grip strength, and rotor rod testing for 12 weeks. So first we'll take a look at the rotor rod. We actually found no difference in rotor rod between the GFP group and the TDP group. So the animals at this stage are not severely impaired, don't have severe motor impairments with the TDP43 cytoplasmic expression in this model system. We then looked at grip strength, and again, we found no significant difference between the two groups, the GFP control group and the TDP43 AAV group. But what we did see was a significant decline in the grip strength in the DNAGB5 knockout with TDP43 AAV compared to controls. Further, we assessed the ability for the animals to produce a normal hind limb splay, and the GFP animals showed an ability to conduct this test normally throughout the entire 12 weeks, whereas the TDP43 delta NLS group showed a progressive decrease in the ability to produce a normal splay and showed a collapsed splay instead. And this produced more rapidly, so there was a more rapid, I guess, collapsed splay developed in the DNAGB5 knockout mouse with the TDP43 AAV. So these pieces of data combined tell us that DNAGB5 knockout exacerbates motor phenotype in TDP43 delta NLS AAV mouse model. So what do all these findings tell us about the human disease? So is there any relevance of the findings from the ALS, from the MND RNLS mouse cortex to postmortem brain tissue from humans with these diseases? So to address this question, we took a look at the late disease mouse cortex findings, and we correlated those with end-stage postmortem human tissue TDP43 proteinopathy datasets. It's a bit of a mouthful. So we took the findings from 6 weeks in this study, the proteomics, and we compared those to Hassan et al. 2022, which was a transcriptomic study looking at frontotemporal dementia versus control. And we also compared these findings to Umo et al. 2018, which is a proteomic study of FTD and ALS frontal cortex. And what we found was that proteins that are decreased in the mouse at late stage disease were also decreased in humans at late stage disease, depicted here by these blue sections of the pie chart. And proteins that were increased in the mouse cortex were also increased in the human brains as well. So shown here in the red in the pie charts. We then looked at the changes in proteins in mouse cortex versus human using the proteomics dataset, and we found two protein-protein interaction networks in common, one related to the respiratory chain complex and another related to calcium ion import. And then the last piece of information, I think, really strongly validates our findings from the mouse study and their relevance to the human disease stages, was this comparison in our modules. So if you remember the colourful modules from a few slides back, black, red, magenta, turquoise, etc. We compared these modules to the modules identified in Umo et al. 2018. So they also used the same technique to look at changes in their proteomics datasets. And for every single module that we identified in our study, there was a highly correlated or a highly similar module in the human study as well, which really reinforces that the findings from the mouse study correlate strongly with the human proteomics. All of the similar modules moved in the same direction as well. So, for example, the turquoise module, if you remember, was decreased in disease in our mouse model, and likewise the turquoise module in the human was decreased. And by happenstance, both turquoise modules are involved in a decrease of chemical neurotransmission proteins. The exception to this is the magenta module. So here the magenta module showed a really high % similarity to the pink module in the human dataset. However, in our study, the magenta module, if you remember, goes up, it increases in disease, whereas in the human module, the proteins go down in disease. I think that also highlights the uniqueness of this magenta module, and I guess the utility of looking at the early disease stages and how they may not reflect the changes at end stage of disease. Okay, so lastly, I just wanted to introduce the web tool. So this was produced a very rich data set of longitudinal proteomics of the TDP43 mouse model, the RNLS8 mouse. And we wanted to make this, I guess, user-friendly. We wanted other people to be able to interrogate the data set and look at genes and proteins of interest from their own studies. So together with Dana, we produced this web tool. It's called TDP Map. And I think I was going to get some help from Sarah to drop that web link in the chat so that you can have a play. And if it's not in the chat, you can type in this web address now if you like to have a look, or you can scan the QR code. And basically, this web tool enables you to input any protein of interest and to pull the data from the mouse cortex or the human data sets as well. So here we have data sets of choice. You have a choice of the mouse cortex or the human transcriptomics or the human proteomics. And then within the mouse data set choice, you can have a look at the changes at each disease stage as well. So it's week 1, 2, 4, 6, and in recovery as well. Everything is completely downloadable. So here in this tab here, data available, whatever you input into this ID, it'll produce a table full of the results, and you can download those results and plot them in any way you like. However, if you don't want to plot them, there are some different visualizations already here that you can explore. So in this instance, the, I guess, default list of proteins are the magenta module proteins. You can see here a log-fold change difference through time of disease. Here, this tab is the volcano plot. It's an interactive volcano plot where you can hover your mouse around the volcano plot and visualize what proteins are increased, decreased over time. And then we have an upset plot, pie charts, and the WGCNA analysis as well. So I really encourage you to check that out and input your proteins of interest to see how they change in disease in this mouse model. Okay, a few last slides to wrap up. Just wanted to summarize the body of work. Firstly, we found that there are very dynamic biological process alterations in the cortex of the RNLS mouse over time. So what we see at the beginning of disease doesn't necessarily always reflect what we see at the end of disease. And those are all sort of illustrated here as different waves. So here we focused on the protein folding magenta module. And within that magenta module, we looked at one of the proteins, DNAGB5. And we found that its overexpression protects against DNAGB5 aggregation. And we also found that its knockout actually exacerbates the disease trajectory of an AAV9 mouse model of cytoplasmic TDP43 disease. We also took a look at the changes in late-stage disease, and we found that there were really strong correlations between both human and mouse. These findings, I guess, give us, a platform to start to look at biomarkers, therapeutic targets, and it also provided us with an idea that this turquoise module is somewhat restorable. So there's a bit of neuronal plasticity. Even at late stages of disease, the neurons are able to upregulate proteins again and partially normalize back in recovery. So it gives us an idea that if we're able to identify the correct therapeutic targets, we may be able to restore the function of neurons. And then regarding our findings related to DNAGB5, I just wanted to place those findings in the context of other recent findings related to chaperones and TDP43. So back in 2019, DNAJC7 mutations were found to be risk alleles for MND. And there are also recent very high-profile papers that were linking different heat shock proteins with TDP43 in terms of regulating their phase transitions, regulating their aggregation propensities. That's HSP70 family, HSPB1, DNAJB2A, together with HSP70, and now our findings, DNAJB5 and HSP70. So I guess these findings regarding DNAGB5 really are a launchpad to many new projects, and I'm really interested in exploring, what is the mechanism of action, what is the therapeutic potential of DNAGB5, what is the relevance of these findings to human disease, and also I really want to know those magenta module proteins in the same place at the same time to work together to combat protein aggregation. I think that would be really interesting to follow up with. So lastly, I just need to thank everybody who participated in this project. It really was a huge body of work, extremely collaborative within our lab, so within Adam Walker's lab, and also with other collaborators. So Dana, our bioinformatician, Mehdi, who led the proteomics, Prachi, Roger, Marco, Albert, and Leszek. And I also finally want to thank the support. So FightMND, I guess, really launched my career in motor neuron disease research by giving me the FightMND Early Career Research Fellowship 3 years ago. It's very nice to be here presenting some of the work that I did as part of this fellowship today, which is very close to the end of the fellowship. Thank you all for your attention today, and I'm more than happy to take questions. Thank you.
Awesome. Thanks, Rebecca. I can't believe that's some of the work as part of your fellowship. You've been a very busy lady, and not only doing incredible research, but also supporting FightMND through selling beanies in Brisbane this year. So thank you so much. It's my pleasure to open the floor to questions. Just a reminder to raise your hand, and we can enable your microphone and video function. If you could please introduce yourself and your affiliations so that we can all get to know each other a bit better before you ask your question. I think while people are getting ready, Beck, a lot of my questions I had were actually on your last slide around DNAGB5, how you think it's regulating TDP43. Of course, is it a potential target? And also wondering, is this something that's specific to the cortex, or do you think that this might also be a key target in lower motor neurons as well?
Yeah, all really great questions. So far the data is looking like DNAGB5 could be a really important therapeutic target. We've done very basic experiments with them. There's so many more experiments to be done to investigate it further. So I'm really interested, how far can we push the system? So if we were to give the cells and the neurons more and more and more of DNAGB5, can we see even greater clearance of TDP43 pathology? And then we have to test to see whether that is associated with improved neuronal viability as well, which we haven't studied any of those aspects in this work. Regarding the mechanism of how it's working, I don't want to pigeonhole it with all other DNAJ proteins and say it's working exclusively with HSP70. Obviously, in the magenta module, we saw that HSP90 was increased, which is a similar system to HSP70 involved in protein refolding. So I think there's a lot to explore there with regards to the mechanism of action. Is it going to many different refolding proteins delivering misfolded TDP43 for refolding and degradation? So I think those are things that I would really like to explore. And regarding the last part, the spinal cord, I actually haven't looked at spinal cord to see whether it's altered in the RNLS mouse in the spinal cord. I will make a note of that, and I should go investigate that. But I believe, if there's cytoplasmic TDP43 there, it's a client of DNAGB5, so they will interact. So it could be beneficial in the cortex, in the upper and the lower motor neurons. Thanks, Beck.
No worries. And I'm going to follow up with another question. And do you think it's specific to TDP43 or other misfolded MND-linked proteins or RNA binding proteins?
Yeah, that's a really great question. There has been a little bit of work in the literature on DNAGB5 and a really, really little amount of work, maybe 2 studies. They looked at FUS, so mutants of FUS, and DNAGB5 overexpression actually increased the misfolding of FUS in that system. So it probably is going to be specific to the protein. It's not going to be, it might not be a generic, one tool fixes all, be personalized.
Great, thanks. Any questions from the floor? Give people a chance to put their hands up.
Rebecca, can I just ask you if there's any evidence of DNAGB5 in other disease pathologies at all?
Oh, that's a really great question. The mutation of DNAGB5 is actually associated with other neuromuscular disorders. So it is an important protein, and I guess the mutation of it is because it's associated with other diseases. It shows that the loss of function of this protein is detrimental. So yes, there is.
Great, thanks.
I think everyone's feeling a bit shy today. Here we go. Is Isabel Deluzzi?
Hi. Hey, Rebecca. Can you hear me?
I can hear you, yes.
Wonderful talk, really, really enjoyable. I wanted to sort of come back towards the middle of your talk where you briefly touched on oligodendrocytes and how you saw that increase in myelination and what kind of, I guess it would be great if you could kind of share a bit more about what you think is going on there. And also, if you don't mind touching on a little bit about why during the recovery you don't see it coming back to baseline. And do you think that that's because it's not really possible to rescue in MND, or do you think something else might be going on?
Right, great questions. Yeah, so we're really interested to see this increase in oligodendrocyte proteins. Also, I think specifically because it remained increase in the recovery animals. There's so many reasons why they could be increased. It could be a protective mechanism. So could be the existing oligodendrocytes that are there are increasing their myelin sheaths in a protective response to axonal retraction or axonal degradation over the time course of disease. Or it could be that there is an increase in the number of immature and mature oligodendrocytes over the time course of disease. And those, I guess, are 2 alternative hypotheses that we do need to follow up with. So sorry, what was the second part of your question?
I think you covered everything.
Oh, okay, yeah. Great.
Okay.
I will also add, so Adam submitted an application to FightMND in the latest round, which was successful, together with Sam, Carly, Emma, Leszek, Marco. It was a very big project to follow up on this oligodendrocyte finding in a lot more detail. So we haven't actually started that project just yet, but basically that finding has opened the door to a lot of questions that we're really interested in pursuing, and we will be shortly. Thank you to FightMND.
Awesome.
Thanks, Isabel.
Great, thanks so much. Doesn't look like there's too many questions from the floor today. Everyone's maybe a little shy, so we might wrap it up there. Thank you, Rebecca, for making the time to present to us today. It was a really fantastic presentation. You really went through all your findings really clearly and really highlighted the importance of them. So if there is any other questions that you think of, or you'd like to get in touch with Rebecca, you can get in touch with her directly, or if you'd like, contact us here at FightMND and we can help us to connect to the 2 of you. So today's seminar has been recorded, and it will be available on our website in about a week's time. So if you know any of your colleagues who weren't able to join live today, please let them know that they can watch it soon. So thank you again for tuning into the FightMND scientific seminar today. We hope this series has been creating opportunities and ideas for our research community. Our October seminar will be held on Tuesday, October the 24th at 12 p.m. Australian Daylight Savings Time. So keep an eye out in your inboxes for details of next month's speaker. So thank you and see you again next time.
Great. Thank you so much, Beck and Sarah.
Thank you.